think I'll get started. So thank you everybody for having me. It's really nice to be here. My name's Jacob Stern and I'm the lead product manager at 10xGenomics for the Visium platform. I'm going to go first and I'm going to do sort of 15 minutes on the latest on Visium and Visium HD. And then my colleague Leilani is going to talk about Xenium. We can take questions sort of interspersed throughout. Maybe we can probably pause after my section and then pause after Leilani's section and then do as many questions as you want during or after. But I'll get started. So quickly to set the stage, we've developed two spatial technologies at 10xGenomics that we see as complementary. There's the Visium platform, which is really oriented around unbiased discovery. It's sequencing based, and so we're able to leverage the really high molecular throughput of sequencing technologies. Because of that, we're able to do whole transcriptome analysis. We've developed technologies for doing this in a 3' based manner, and also whole transcriptome probe sets for working with more challenging and even clinical samples. And because the barcodes are in fixed areas, we're assigning transcripts to multi-micron areas on the slide. With the Xenium platform, it's imaging based, so we're able to get really precise localization of individual molecules down to the nanometer scale. Because of that, we're able to more precisely segment cells, and so we've invested quite a bit in improving cell segmentation. It's a continuous process to really be able to do single cell spatial analysis in a way that's native to the single cells. But the trade-off of doing this in an imaging-based way is that from a molecular standpoint, it's lower throughput. So we're doing hundreds and pushing to thousands of targets relative to the whole transcriptome. I'll talk about the Visium platform, and then Leilani will talk about Xenium. So we first launched Visium in 2019 after acquiring a company called Spatial Transcriptomics out of Stockholm in 2018. I'm pleased that we've been able to switch this slide from publications and preprints to just publications. And as of our last sort of formal count, we're up over 400 publications at this point, some of which come from people who I think are in this room or certainly at this institution. And it's exciting to see how this is sort of compounded over time. And as the technology has sort of been out there for longer, we're starting to see more and more research across more and more disciplines that are able to take advantage of this technology. I'm going to start by talking about the CytAssist, which is an instrument that we introduced in the summer of 2022. I think the core here has 3 CytAssists, and this is one of the best Visium cores in the entire world. Honestly, you guys are very lucky to have it here, and they're really proficient at using this to drive research. Well, we designed this machine around making the overall Visium workflow simpler. When the technology first came out, one of the things you had to do was section and place tissue directly onto the barcoded Visium slide substrate, which of course introduced challenges in actually figuring out how to do that. The high stakes of somebody who's used to working with 25-cent glass slides, working with this multi-$1,000 slide, and actually having to cut down tissue to work with it. So we do fun code names when we develop projects at 10x. This one was Foreman because it's like a George Foreman grill that makes a slide sandwich. I'll go through that, basically why that works in a second. But really, we designed this to plug more directly into standard histology workflows, allow people to do their tissue prep the way they always do their tissue prep, and provide access to real-world samples where perhaps you can't get the whole block, you can only get a slice of tissue on a slide, or you're trying to work with tissue microarrays that are more challenging to cut down. One of the interesting things that we learned along the way is that the CytAssist actually delivers higher quality data than the direct placement approach. The lead developer on the CytAssist came from the microfluidics department at 10x, and we really engineered the system for precision. And we'll talk a little bit about why that matters for HD in a few slides. And of course, because we're able to plug into standard imaging workflows, it allows people to do things with H&E and IF, et cetera, that's more standard, or even in many cases, as you heard in the talk right before lunch, trying to go off book and do some fancy things upstream of the CytAssist, which in our experience has more rather than less tended to work. So briefly, how the CytAssist works is you think about how this could fit into your own experimental workflows. Tissues is sectioned onto standard glass slides. You go through sort of a standard imaging process, generate your high resolution image. And then here's where we get that George Foreman grill concept. You take 2 of the standard glass slides, and then the Visium slide with 2 capture areas. And the CytAssist brings the sample very close to the Visium sample, and the permeabilization and barcoding step happens on the instrument. And while it's going, the instrument also takes a picture of the interface, and the software that we've developed basically takes the image that was taken on the high resolution image, the sequencing data that comes off the sequencer, and then the image on the CytAssist that sort of aligns where are the fiducials on the Visium slide relative to the tissue, and brings it all together so that you can put your high resolution image in the same coordinate space as your sequencing data from the Visium slide. I'm going to walk through a couple of examples of how that's worked in the field. I apologize, I did a red eye last night. I didn't have time to pick something specific to Michigan, although there are many, many, many examples here. One example from the original inventor, Joachim Lindeberg, who developed spatial transcriptomics in the first place. He did a one-year sabbatical in Australia and developed some sort of tight collaborative relationships down there. And they're running a correlative study on a breast cancer trial, where they took a couple hundred samples of breast cancer patients, and they're basically trying to produce a breast cancer atlas. And they did all of the histology and annotation at the clinical sites in Australia, and then hardcover slipped these samples, shipped them to Sweden, where they did the molecular biology. And so the CytAssist enables such things to work. I know Liv with the core does a lot of work with samples that are shipped from all over the place here because they run the core so well. And having the CytAssist actually makes such a thing possible. Another one, which Liv might not love this example as much because it's more of a risky one, but Hanli Ji is an oncologist at Stanford. I was in his lab in November, and he was telling us about a project he was running on gastric cancer samples. And he had these samples that were already on slides in a drawer somewhere for like 4 years or something like that, hardcover slipped, necrotic. But these were the only samples they had from the specific cohort, and they just needed to run them. And so they said, let's give it a try. And they were able to actually get data from these samples. And I can see Liv sort of making comments in the back, like, why is Jacob talking about this? But, you know, buyer beware for something like this. But working with actual clinical samples that are on slides is something that's possible. So when we first developed Visium at 10x, we produced Visium slides that had 55 micron spots, 5,000 of them in a 6.5 millimeter capture area, or about 14,000 of them in an 11 millimeter capture area, with 100 microns from center to center. One of the wonderful things about serving the research community like yourselves is that people are not shy in telling us what they want. And one of the things that came back loud and clear almost immediately was, please make this higher resolution. We want to be able to think in terms of cells. And so we have been working on this for a long time. It's an extremely challenging development and manufacturing problem, but we have finally gotten there. And so this is what it looks like. These are serial sections of the same colorectal cancer sample. And what you have on the left is what this looks like on the CytAssist with the 55 micron product, and then on the right at Visium HD. So what have we been able to do here? We're maintaining that whole transcriptome analysis for human and mouse. We have probe sets that are about 18,000 genes in human and 19,000 genes in mouse, mostly threefold coverage for paired probes to maximize the chance that you can actually read the gene you're interested in. We've now moved this down to single cell scale in a 6.5 millimeter capture area, which is where we've started. You have a continuous grid of 2 by 2 micron squares. So rather than having 5,000 spots of 55 micron within that area, it's now about 11 million distinct barcodes in the format of 2 by 2 micron squares. It's built for FFPE because it's leveraging the CytAssist, the same benefits that we were talking about before, and using the CytAssist to do work with Visium, transfer one-to-one with working with Visium HD. And again, broad access, same thing. The enhanced histology, same thing. The high quality data, same thing. Because it uses the same instrument and the same principles, and the only change we're making is on the barcoded slide that we're using towards the end of the workflow, you're able to still plug this into your standard histology workflows and work with the same sort of suite of samples that you could use for Visium for the last number of years. And then we invest heavily in our informatics tools at 10x. Loop Browser in particular has gotten a substantial upgrade to work with this data. And if you haven't already, I would recommend playing with the data that's available for download on our website. I think we have 4 public data sets now available for Visium HD, and you can download Loop Browser for yourself and download the loop file from those data sets and play around with it and get a sense for what the data looks like. I always find that a live loop demo for myself is the best way to familiarize myself both with a sample and then also sort of what's possible with the technology. So I'll sort of step through a few of these points one by one. I like this format of slide. It's sort of the library of beautiful Visium samples. We do try and test across a broad range of real-world equivalent sample types. We'll sort of buy samples from a biobank that have often unknown provenance and run them to see. So we have a range of healthy, diseased human and mouse tissues with differences and sort of the makeup of the tissues to try and test across a dynamic range to make sure that our protocol is as universal as possible. And I'll just step through some of these in blown up formats so you can get a sense. For anybody who's used to looking at the sort of 55 micron data, where you can get a sense for the degree of precision you can get out of the clustering here, and the level of variation, heterogeneity, and tissue architecture that just pops up through the unbiased clustering. So human colon and colon cancer, lung cancer, prostate cancer, tonsil, and also mouse embryo. Mouse embryo is really fun to work with because it's so spatial and so varied, and you can really pull a lot of details out, even with just the unbiased clustering. We want to test to make sure that sort of performance across a wide range of prep conditions is also maintained. So here you can see an experiment with a bunch of different conditions upstream of going to the CytAssist, ranging from freshly mounted to stored for 6 months at RT, at 4 degrees Celsius, hard cover slipped, and basically they're all equivalent. So the flexibility you have upstream is there, and it's sort of robust to a bunch of stuff upstream of actually running the CytAssist. One thing that we thought about really carefully during development is how to maximize the actual functional resolution of this assay. Because it's one thing to have the 2 by 2 micron squares as the feature size, but if you have molecular diffusion, if you have image registration errors, whatever it might be, things that were immaterial when we were working with 55 micron spots all of a sudden become quite material. And so we focused quite a bit on how to maximize the actual resolution of the full assay as an integrated thing. And the CytAssist, it turns out, is really important for this. So we obviously started developing Visium as what we call a direct placement approach, putting tissue directly onto the Visium slide. And there's just fundamental issues with that approach in terms of where molecules end up. When you start permeabilizing, the molecules can end up in solution, they can move laterally. As they go through tissue, they have a chance to move around. And then also if you have distance between spots, then molecules, when they land on sort of blank glass, can have a chance to move around until they find a landing home. With the CytAssist, again, sort of led by somebody from the microfluidics department, we've done quite a bit to do controlled mass transport in that permeabilization and barcoding step. And because you have a controlled environment that's closed, we sort of minimize the amount of diffusion during that permeabilization step. And we actually built this into our requirements as we were developing the product. We designed these systems where you have known markers that you can cross-reference against the image from the same sample. We're basically testing how many of the molecules we'd expect to be segregated to a specific area end up actually there. So here you can see a demonstration of what this looked like during R&D as we were sort of optimizing around this effect. And here you can see the result. These images were taken straight out of Loop Browser 8.0, where you're able to do multi-gene or multi-signature analysis at once. And you can see across a range of tissue types here, sort of markers that are supposed to be very spatially segregated. And you can see the precision with which you're able to see these, even for single gene markers. The line of the, I think it's the LYZ1 marker in the mouse small intestine in particular, where it's very confined. The epithelial compartment is really cool. In terms of sensitivity, we've been able to basically maintain the sensitivity that we had for the CytAssist assay at 55 micron resolution per sort of tissue area. Generally, it varies by tissue type, but HD is going to end up being modestly more sensitive than what you're used to if you've been using Visium on the CytAssist so far. Here you can see a distribution on a heat map of UMIs per 8 micron bin within this particular colon cancer sample. In this case, I think this sample was sequenced to around 400 million reads and was about 46% saturated. So there's further room to run if you want to sequence deeper on something like this. And you can see that you do see basically the number of reads are, as you would expect, different by the area of where you are within the tissue. And in the hot regions, we're getting upwards of 1,000 UMIs per sort of 8 micron bin, which is approximately cell equivalent, though not exactly a cell. One of the cool things about this, that again, this becomes more powerful as we increase the resolution of the assay, is getting imaging from the same sample. The type of analysis that we were doing around the actual resolution is really only possible because we have ground truth from an image from the same tissue section. And here you can see why that matters. On the left, you can see section 1, and on the right, you can see section 2, which are serial sections. These are 5 microns apart. On the left, you see the single plasma cell that's present in the H&E, and you're picking up J-chain as a signature for that plasma cell where you'd expect it to be, again, very closely confined. On the right, on that same section, the plasma cell is gone, so is the signal. So at this level of resolution, things do actually move as you go through a tissue block. And so we think it's really important to have that imaging from the same section. And stuff like the multimodal cell painting that was talked about earlier can become really cool with this technology as you start to pair that sort of high content imaging with the whole transcriptome sequencing at resolutions that are at least somewhat comparable. Something else that we've done internally, and we're sort of preparing a preprint to put forward using all of our technologies, Visium HD, Xenium, and Chromium Flex from the same FFPE tissue block, is starting to work with single cell data and Visium HD data and doing deconvolution on this now higher resolution continuous coverage canvas. Here you can see that basically tumor signature that was uncovered in Flex data, basically at actually the cellular level, is sort of matching to where you'd expect it to be in the unsupervised clustering from the Visium data. And then we can start to do stuff like this. So here the red is the tumor signature. And we're able to draw these gradients basically around that tumor and then think about neighborhoods and what's the gene expression information that's happening within 15 microns of the tumor signature. How about between 15 and 30 microns away? How about between 30 and 50 microns away? And you can see just a couple of examples of markers that sort of vary in their expression based on the distance from tumor that it is. And because we have the level of resolution, we have the level of precision, and we have the continuous coverage, you can actually start to do this and really dive down into microenvironments and try to understand what's happening. So Visium HD is now available. I think Liv has already gotten started with running. So, and again, you have access to one of the best Visium running cores in the entire world. So it's an amazing resource to have here. And just to reiterate, unbiased spatial discovery from tissue sections, resolved at single cell scale, where we're able to do this on the order of single cells. With data quality you can trust because we've really engineered and optimized this assay to give you the functional resolution through the CytAssist and other aspects of the assay that you'd want from a high resolution spatial assay. So with that, I think we can probably do some questions. I know there are a couple of questions that I've already been primed for. So one is, what about the larger capture areas, the 11mm capture areas on HD? There are a few things where basically we didn't want to gate getting HD out the door until we had full feature parity between HD and the 55 micron resolution. And so now we're building everything back up on the HD slide substrate. So the first priority that we're working on is actually a 3' based assay on HD through the CytAssist to get access to non-human, non-mouse species, sort of hackable for things like reading CRISPR guides, compatibility with long reads if you so chose, I'm particularly excited about what that might be able to do for sort of spatial VDJ analysis, particularly at the high resolution if you pair it with long reads or sort of a nested PCR type approach. And then we are also working on sort of larger capture area formats. I have learned my lesson the hard way about coming out with timelines, so I'm going to hold myself back from doing something too concrete there. The other one was asking about sort of NGS-based multiplex protein analysis, so similar to CytSeq, single cell realm, because we do have an assay available on the CytAssist right now with the 55 micron resolution for doing this in a fully supported manner. The difficulty there is that the CytAssist slides have two different capture sequences within each spot. You have poly-T, which is there to capture the RNA probes, and then capture sequence 1, which is used to capture the antibodies. You end up making two different libraries. The Visium HD slides are just poly-T, and because each square is smaller, it's sort of hard to engineer in two things. We might be able to do it down the line, but basically we will have to rethink the NGS-based protein approach from the ground up. I think we all have bets on the over-under on how long it takes someone to throw a panel of total CK antibodies and see how it works. But it's something we're definitely thinking about around how do we enable such a thing. But that's sort of further out in the future than a 3' based assay or a larger capture area format. So happy to take any other questions. In the back. The position of HLA-KIR, I guess in what context? I have to look. I think we had them in the version 1 probe set for Visium FFPE, and then when we launched Flex and did the sort of 3 probes per gene, and we removed them because there's some ambiguity on whether the probes that were targeting individual HLAs were actually hitting those individual HLAs or more of a class. And so I'm pretty sure we did remove them. The probe sets are available for download on the website if you want to check. And if you wanted to spike them back in, we do have a tech note that's quite popular that details in a great degree of detail how to design probes. If you wanted the specific ones from the V1 probe set, you could just order those. And there's super detailed instructions on how to order oligos from IDT or your oligo provider of choice, the assay conditions to put them back in. So we've tried to make that as self-serve as possible. It's not all the way self-serve, but as much as we can.
What about, have you done any cell segmentation on these to see if you can kind of get those into that area or go down to a lower 4x4, 2x2?
Yeah, the question is on cell segmentation, and the short answer is yes. It's another one where I think the community is going to get there before we offer formal support. I've actually already seen it where somebody's taken a high-res image and then the 2x2 micron data, thrown it into CellPose and used that as a mask to basically draw custom binning. Because what you'd want to do is get as close as you can to assigning 2x2 micron squares to sort of cell-like boundaries. We do have an analysis guide on our website if you want to search for it, where we did this with a nuclear segmentation, basically taking anything that's under a nucleus as extracted from the paired H&E. So we've sort of given some clues and a starting point there, but I think there's going to be an explosion of methods for doing this very quickly with various flavors, depending on what you're looking to do. And one thing we noticed, by the way, as we do that is as you throw away 2x2 micron squares that aren't under cells, the counting metrics actually go up, because as you'd expect, the density of reads is greater where you actually have cellular material that has on it versus the average of everything that's on the tissue. The question was this automated, and then could it be exported to third-party tools? This was done by an in-house computational biologist who's sort of working on more of a demonstration. I think this was actually done in third-party tools. When we publish that preprint, the methods section will contain all the instructions for doing it. We shared data early with the makers of Seurat and Spatial Data, a few of these packages, STLearn, so that they could get a head start on updating their packages to work with Visium HD data. So I think that's in reasonable shape at this point. It's definitely true that the data packages are substantially larger. And so it's something to think about in terms of the computational resources you'll need to run the same types of analysis. Cool. One more in the back. Necessity for what? Sorry, I didn't hear. Yeah. Question was 20 versus 40x imaging for HD. I think probably the unsatisfactory but true answer is depends on your question. The higher, basically the data from the sequencing data now can sort of go toe to toe with higher resolution imaging. And so you'll get more for it there. The sequencing data, though, is sort of living at that 8 micron-ish resolution. So if you're doing 40x, your imaging data is going to be substantially higher resolution than your sequencing data equivalent. If you're trying to use the imaging data as a first-class part of the analysis, which is awesome. I think a lot of the coolest stuff that's coming out with spatial transcriptomics really treats the image as a first-class part of the data package. Then 40x can really give you something, but I wouldn't say it's required. Thank you very much. I'll pass it off.
Perfect. All right, thank you, everyone. I appreciate the opportunity to speak here. I know that I mention this every time I come to give a talk here, but I actually did my postdoc here, and it's always really fun to come back and be able to talk to you guys about our technologies, especially because when I actually was doing my postdoc here, I was working on a manual version of the Xenium protocol for some of the workflows that I was doing. Now, whenever I talk about Xenium, I know that Jacob put into context how important spatiality is when you're looking at biological questions. It's something I noticed as well from the posters that I was looking at earlier, where this is pretty much a very heavy topic, it seems, for this group. But I really like to show these animations, and I like to show a couple of the next slides that I'll be presenting because it really puts it into context. So what you're actually seeing here is that each of those dots that gets collected into that UMAP, looking at that clustering pattern, is actually going to be an individual transcript that you're looking at instead. So we have very high resolution when we talk about Xenium. We're looking at the transcript level, pretty much down to 30 nanometers in the X and Y coordinates. Like I said, I always like to start with actual data. So what we're going to see here is going to be this morphological readout looking at this whole mouse embryo. This is a post-Xenium H&E staining that was done. Xenium is the only true non-destructive methodology out of any of the existing in situ platforms. So you have that capability of doing post-Xenium H&E, post-Xenium immunofluorescence, theoretically even post-Xenium CytAssist, if that's something that you're interested in as well. And so what we're going to do is we're actually going to zoom in on a specific region of this mouse embryo. We're going to take a closer look at the kidney of this mouse organism. We're actually going to continue zooming in. So if I begin to look at this, I can kind of begin to understand, we have the cortex, we have the medulla, we have all these different regions, but once again, the power of Xenium lies in being able to get down to very high levels of resolution. So we're going to zoom in on this particular area, focusing on common structures associated with the maturation of the developing kidney. We can see areas like progenitor cells located here. These are maturing into different cell types as we go through. We can see our tubule areas, we can see our ureteric bud. And so we're able to get to that very nice morphology that a lot of people really need to be able to understand their biological questions. Now, what I'm going to do is I'm going to overlay additional stains. And so we recently rolled out our multimodal segmentation cocktail for Xenium. And what this does is it provides additional information about cell boundaries. So what you're seeing here in red are going to be stains for ATP1A1, E-cadherin, CD45. In that orange color where you have that overlap with the red, you'll have your cytoplasmic RNA, so an 18S, to be able to look at cytoplasmic boundaries. And then in green, you're seeing alpha smooth muscle actin and vimentin staining to get to further cytoplasmic stains of different cellular compartments. We combine this with DAPI, and you're really able to get to that cellular morphology via the staining. And then you're able to detect those cell boundaries, find the demarcation between cells. And this becomes important because when we're beginning to detect transcripts with our technology, which is what you're showing here, where we can see your RNA that has been labeled with these fluorescent oligos, you can actually locate these RNA transcripts with subcellular precision, ultimately overlay that onto that cell morphology image that I showed earlier, and then understand how those transcripts are assigned to individual cells. And that way you can put together that information of RNA content with each individual cell and really understand what is happening within each of these cells. So you can see how you have your individual cell populations where you have nephron progenitors, you'll have these ureteric epithelia, and so on and so forth, to really be able to understand things down to that subcellular level of information. You know exactly where each cell is living within the tissue. And so if we come back to that H&E image that we started with, which is that standard pathology image, you can now overlay this data with that cell clustering. So being able to understand what each cell is, where it is within the tissue, and what it's doing to much higher degree of information based on that single cell expression characterization that we did. Now, the next question that I get from a lot of users is, how do we do this with our Xenium technology? And this is based on our Xenium chemistry. So what you're seeing here is the capability of sectioning onto a relatively large tissue area and then combining it with our probes. This is standard histology. So if you're working with fresh frozen samples, you would do your standard washes to remove any excess OCT, and then a permeabilization step to allow our probes to penetrate. If you're working with FFPE samples, standard deparafinization series, a G-cross-linking step that's analogous to antigen retrieval, and then our probe hybridization. We do a very similar approach to what we do with our Visium and even our Flex technologies, where we'll have a left-hand and a right-hand side probe that need to come in and bind specifically to your target RNA. And so what you'll see here is the ability of both of these having to come in and bind to recognize each of those targets specifically. Then from there, we have a highly specific ligation event. When I say highly specific, we're talking down to the single nucleotide level. I already have users that have designed their own probes for gene fusion events or isoforms. We're working with users currently that are designing probes, either single nucleotide variants or single nucleotide polymorphisms, and even beginning to look at things like clonotypes as well. From there, you've now created the circular probe, so we take advantage of that and do a rolling circle amplification. And what this does is it generates multiple copies of this gene-specific barcode that you're seeing here at the back of this probe. This comes into play when going onto the instrument, because then what will happen is you have an automated fluidics component, which will wash on different fluorescent labeled oligos that will bind to each specific gene. From there, it will move the slides over to the imager. Once an image is generated, it moves it back. And so it's doing this in a cyclic manner. So in my previous cartoon, I showed one single probe. We're actually doing multiple probes per gene based on whatever the existing information there is on the expression of those genes in this particular tissue. And then you're working with all of your genes at once. Currently, we're at a plexy of about 300 to 500 genes, but we'll be increasing that very shortly. And then on the right-hand side, what you can see is the ability to do what we call internally inline decoding. So working with code words or those individual optical signatures to know where each gene is being expressed at each cycle of the instrument. Now, I mentioned that we do multiple probes per gene. We work with multiple genes at once. And what a lot of our users want is that turnkey solution. And so what we did is we created these pre-designed and validated panels. You can see that we have 7 human panels, 2 mouse panels present on here. And so these panels provide this conjunction of genes that we've determined based on either existing single cell and Visium data. We've worked with different leaders in the field, and we've also mined a lot of the scientific literature to make sure that we're covering the genes that people need. We just recently added our human immuno-oncology panel, which is our first application panel, and that's been of some interest to our users. But one of the things that we found is that one size fits all really fits no one when you're doing this type of workflow. And so what we've done is we've allowed for customization, so you can add custom content to any of our existing panels, currently up to 100 genes to any of those. The other thing is you may be looking at this list and saying, okay, those are good, but those don't really apply to what I'm working on. So we do have the capability of doing full custom as well, up to 480 genes currently. And we've even taken the approach of doing things like advanced custom. So working with things like different mammalian species, things like xenografts, plants, and a broad variety of other applications. Does anyone in the room know what Aedes aegypti is? See one hand up and a couple hands in the back. So for those of you that know this, this is a mosquito that is going to be the vector for things like Zika, dengue, and a couple of these other diseases. And so that shows that we have the capability of doing things like insect workflows as well, if someone is interested, let's say in Drosophila or something like that. Now, if you have any questions about that advanced customization, this is why we're here. So please come find me after the talk. I'm always happy to talk to you about what best approaches are. I do want to highlight the fact that we do have that immuno-oncology panel, since this has been something that users have been asking for basically since we released the Xenium analyzer. So we do have the capability of doing a lot of different cell phenotyping, whether you're looking at different tissue compartments or whether you want to go really into the nitty-gritty of your individual immune cells in particular interactions. And we've seen this, for example, where people have performed spatial niche analyses to be able to distinguish different spatial cell compositions between the tissues. So they can basically determine where you have these individual niches and where you have the expression within individual cancer samples overall, and ultimately be able to do their immune cell profiling within those tumor microenvironments. If you do have interest in being able to build your own custom panel or maybe add on some custom genes, what we've done is we've actually created the Xenium panel design tool, where you can basically upload your genes of interest into this particular algorithm, and it will design to those genes of interest, making sure that it works well with any of the existing panels, if that's something that you're interested in, or it will be able to look at the expression patterns based on reference data that you provide. If you have questions about this, I know that Liv highlighted the importance of being able to have a very thoughtful process as you're doing this design. Please let me know. I regularly walk people through using the Xenium panel design tool, and it's something that I've found a lot of users have really liked because they can potentially put it to the side for a little bit, think on it, come back, change out their genes, and then they have the perfect panel for their workflows. I do want to reemphasize the fact that we are doing this advanced customization, and a lot of these things are only capable because of the way that we've designed those probes with our Xenium chemistry. So being able to look at things, as I mentioned earlier, like isoform fusion events, things like that, or being able to look at the TCR, BCR sequencing or express single nucleotide variants. We also have a lot of users that have been designing things like xenografts. So we've been able to design probes to each of the specific species in that and be able to understand, okay, based on these housekeeping genes, the cell that you're looking at is a mouse cell versus a human cell. And that becomes very of interest to a lot of our biologists. And then being able to look at things like exogenous sequences. So potentially being able to look at things like bacterial or viral infection sites, or potentially you're working with a mouse model that has a reporter gene. Being able to even just look at that GFP localization can become important depending on your biological question. I mentioned earlier that Xenium is the only non-destructive methodology out of any of the existing in situ platforms. So we have people that are regularly doing that post-Xenium H&E. I've even heard users such as Liv saying there's no reason not to do a post-Xenium H&E if you have that interest in looking at that tissue, because it gives you that correlation of that histology. We also have people that are doing immunofluorescence currently, so being able to look at proteins to get to a multi-omics workflow with where we stand today. And I've had users that have been working with multiplex immunofluorescent platforms already post-Xenium. So not just being able to do your standard IF, but being able to potentially run a Lunaphore or a PhenoCycler post-Xenium is something that's a capability as well. And lastly, as I mentioned, potentially you run your sample and you run into the scenario where you get an unexpected result with your particular panel. You can now go back to that same exact tissue sample and interrogate it using the Xenium slide as a donor slide on that CytAssist instrument, and then being able to look at this using Visium to get to whole transcriptome information. That way you know exactly what is going on in your particular sample of interest. Now, the last thing that I want to focus on with my talk is where we're going and kind of what things are coming up. I know that that's been one of the requests that we've had from a lot of users. So I did mention earlier that we have our multimodal segmentation cocktail. This began shipping towards the end of Q1, and we have a lot of users that are really excited about this. As I mentioned earlier, it's a series of antibodies and RNA markers that really allow you to be able to define the boundaries of your cell and have better cell calling, better transcript calling in terms of information. This has been strongly validated with human and mouse tissue. However, if you have questions about compatibility with your own sample types, let me know. I was looking at this recently for one of my users that is doing pig xenotransplantation models, and it's something that out of all the antibodies I mentioned earlier, only the CD45 seemed like it wouldn't work in their particular tissue. So it might be something that would be of interest to other users. I'll also talk about our increase in plexi. So we will be releasing these 5,000 plex panels towards the midpoint of this year. We will also be working on inline multiplex proteins, so being able to have barcoded antibodies and your transcriptome readout at that ultra high resolution in the same exact workflow. And then we will be offering additional capabilities in terms of plexi towards the end of the year as well. The first thing that I want to talk about is our 5,000 plex panel. This is something that has been of interest to a lot of users because I think one of the things that we've run into as scientists is the fact that we always need more data to corroborate what we're trying to look at. And so being able to have these standardized 5,000 plex panels for both human and mouse is something that we're releasing towards the midpoint of this year. It's something that you will have capability to do some level of customization, so you will still be able to add on 100 custom genes if your genes of interest aren't listed in those 5,000 genes in this particular panel. And then the other thing that I want to highlight is the timing of this workflow. So currently we're at about 2 days on instrument with our current plexi of about up to 500 genes. This is something where with our 5,000 plex panel, we're not increasing this time linearly. We've made some modifications to the chemistry. You will still be using the same instrument though. And this is something that really allows people to get to their specific biological answer in a shorter amount of time. So being able to get down to 6 days runtime, about a day and a half prep time of the tissue. So it's not something that you're substantially increasing and having to run one sample for months on an automated fluidics workflow. We do have some sample data associated with this, and this is something I'm really excited to see more of. I'm just showing some of the human data here, but like I mentioned earlier, we also have this exact same panel available for mouse. And the other thing that I do want to highlight is the fact that we will be additionally increasing plexi in different ways. We know that some people want to be able to look at either 1,000 plex or 2,000 plex within their particular panels. And maybe they want to be able to run that all in the same workflow using protein as well. And so what we'll be doing is we'll be using these barcoded oligoconjugated antibodies in the same run so that you're able to do this all at once. And so I'll show an example of this here where you're seeing this 20 plex protein plus 1,000 plex RNA panel that was run all at once on one particular tissue slide. And so in this particular case, you're seeing 4 different antibody stains in one tissue. These are 4 additional stains. And then this is the result of the RNA clustering that was done using that 1,000 plex panel. So being able to do multi-omics at once to be able to get to higher levels of information from one individual sample. So with this, I'm actually going to close using the same slide that Jacob started with, where I know that we get a lot of questions from users as to when they would use either Visium HD or when they would use Xenium. And so I usually give the example of a couple of researchers that I work with, where they're both in the same department. One of these researchers is looking at racial disparities in pancreatic cancer. In her particular case, the existing markers that are known don't work in her patient samples. And so because of this, she's turned to Visium and now Visium HD to be able to really interrogate and understand markers that are more indicative of cancer progression in her particular patient samples. However, her department chief is someone that's really interested in Xenium. He's also looking at pancreatic cancer, but he has particular signatures of oncogenic drivers that he's interested in, down to that single nucleotide level. And so because of this, he really wants to use Xenium because that will allow him to look at these particular single nucleotide variants in his samples of interest and then be able to further interrogate how that is looking in these different samples taken at different time points. And so with that, I'd like to say thank you for your time. This is our local team. In case you have any questions, please feel free to reach out with emails or we'll be around afterwards to take questions as well. So thank you again. Any questions from the room? Repeat that? I'll have to get back to you from our bioinformatics team. One of them were the ones that created that, and they've done that for a lot of our technologies. People have really loved those animations. I have a similar one for Visium that just kind of puts that spatial biology into context. So repeating for the room and for the people online, the question is, when is the release time point or time frame for the 5,000 plex panel? Our anticipated release date is towards the midpoint of the summer is what I've been told. So probably kind of towards that midpoint between, the half points of the year is what I've heard thus far. But I think they're going to hang out for a little while. So if you have other questions you want to ask, I think the team will be available. All right. Thank you, everyone.